My name is Niclas Hansen, and I'm a PhD student at the Center for Biological Sequence Analysis at the Technical University of Denmark. And throughout the last 6 months, I've been working on predictive pharmacogenomics with Professor Russ Altman here at Stanford University. So I'd like to make sort of a short summary of what I've been doing over here. And the general field is predictive pharmacogenomics. And the reason for looking into pharmacogenomics is that most medications do not have the same effect on all patients. So some patients might experience side effects and others do simply not respond. And most of our knowledge of such drug-gene interactions between a drug treatment and the genetic makeup of the patient is anecdotal. So we don't have like hard data or any high throughput data that allows us to analyze this on a large scale. So high throughput studies have come forward in the most recent years, and the state of the art is using genotyping arrays that allows us to measure up to 500,000 or even 1 million SNPs or small mutations at the same time. So with that amount of data, we're actually able to capture almost all the genetic variation in a single patient in a single experiment. So this sort of leads to the needle in a haystack problem, because we only have a few set of patients and we have a lot of different potential interesting variants to look at. So that's why we need to find ways to integrate all the knowledge we have about previous drug-gene interactions from all the studies that have been carried out throughout the last 50 years or so. I have chosen to take an integrative systems biology approach where we basically use the structure of the cell, so what proteins interact within the cell, to integrate all the data we have from high throughput studies and all the anecdotal information we have found over the last 50 years. So the goal of my work here at Stanford is to develop a pipeline that allows us to take one drug, its structure, and a set of indications that we believe that this drug treats, and then rank the entire genome in regards to whether we believe that that gene or the best ranking genes would be relevant to this drug-gene interaction. We have developed such a pipeline, and it's based on 1, a human protein-protein interaction network, 2, drug-target interactions from drug bank and structures from drug bank, and 3, pharmacogenetic drug-gene interactions from the PharmGKB resource here at Stanford. And it has a simple data structure that I'm just going to draw. So, first, if you have a drug and you have a gene, you take the gene and you make a pull down. So that means we take the protein corresponding to that gene and look for interactions in our protein-protein interaction network. And then we go on and try to say, well, this sub-network tells us a lot about the function of this gene, because normally things interacting with each other in a protein-protein interaction network tend to be involved in similar phenotypes. So the next step we do is we look for drug interactions to this network. So we imagine we have a drug here, a drug here, and a drug here. Now, we take the drug that is our input drug that we were querying about, and we compare the structure and the indications of that drug to all the drugs that interact with this network. And that tells us something about how likely it is that the drug that we're looking at is interacting with the gene which is the origin of this pull down. So based on this data structure, we calculate 4 features, and these 4 features are then put into a modeling system, and we train a logistic regression model that allows us to predict whether a gene is involved or not involved in a given drug-gene interaction. We have applied this to a set of different high throughput experiments. So we have taken high throughput experiments and taken their top hits, sort of taken the haystack, and then we have ranked the genome and said which of these genes that are associated in this study turns out to actually be relevant in terms of the molecular biology and the drug-gene interactions that we already know. The approach has shown very promising performance, and this means that in the future, pharmacogenomics scientists will be able to take their DNA microarray studies or their genome-wide association studies and then use our approach to integrate knowledge about the cell functioning, the molecular cell biology, and prior studies of drug-gene interactions. That is sort of the first step in filtering the haystack, which would leave them with maybe a haystack, well, the hundredth of the size of this one they started with. And then they could go on to do functional experiments on their best hit on that haystack and hopefully find the needle. And that's it. So, if you want to know more, please go to pgxpipeline.stanford.edu, and you can feel free to contact me at nth@cbs.dtu.dco. Thanks.